with that as a brief introduction, I'm going to introduce our first speaker, Dr. Marla Spivak, and I think most people know Marla, but I'll give a little bit of her background here very, very quickly. So we had dinner with Marla last night, and she mentioned to me that she started school at Prescott College in Arizona. So she was there for about 2 years, I think, and then the college went bankrupt. So, not because of Marla, but obviously it went bankrupt. And now I'm thinking about it. She mentioned she chose Prescott College because of there was not a camping major, but there was a real emphasis on the outdoors, and she obviously is passionate about that. So basically, when the college went bankrupt, she needed to find a place that would actually take her credits, right? She could transfer her credits to another university. She looked everywhere, and I guess one of the few that would actually take her credits was Humboldt State. So she ended up getting a BA degree in Humboldt State, and then from there to Kansas State, where she did her PhD, some tremendous work in Costa Rica, working on Africanized honeybees. And then she did a postdoc at the Center for Insect Science at the University of Arizona. And ever since then, probably in 1993, she started at the University of Minnesota, where she has a spectacular program. She is the Mackenzie Endowed Chair there that she holds. She is also honored. So in 2010, she received the MacArthur Fellowship, which is one of the highest honors that we can give scientists in the United States. And for those that don't know, it does come with a $500,000 payout, basically, that goes to Marla over a 5-year period. So I guess she's in the last year of that incredible honor, I guess is the best way to say it. Her work is phenomenal. She does very fundamental work. She publishes in neurobiology and animal behavior and evolution. But I think one of her real strengths is to take that information and translate that into presentations and into programs and into workshops so she can relate to an audience such as this. I'll say one more thing. There is a new bee and pollination center that is being built at the University of Minnesota. And I think without question, I am sure she is the driving force behind that. So with that brief introduction, let's please welcome Marla.
Okay, that was, thank you, Mike. Overwhelming of me, but true about Prescott College, and I was not responsible for its bankruptcy, but I will tell you that the president of the college at that time had put his faith in God to bring the school out of financial difficulties, and God didn't pull through. It's a whole different story. So I'm going to talk to you today, general talk, and then I'll move into some of the research that we're doing. And thank you so much for having me here and for everybody showing up. This is a great conference. We need to protect our pollinators, and there is a big movement nationwide, as Mike was saying. It's awesome to see what's going on across the United States and across Europe and Canada, and really the public awareness that has been drawn to the plight of our pollinators. And of course, this drawing comes from Minnesota, and that's where I am, in the upper Midwest, and so a lot of it's corn and soybeans. So a lot of the focus that we have is a little different from what's going on in California, but I think the principles apply the same across the board, and that is that our bees need good, clean food. Just as we want good, clean food, our bees need it also. So that's kind of where I'm going to start with this. And I also want to bring up in general that it's just more than honeybees. And I know a lot of beekeepers are in the audience, and I think beekeepers are aware that that's one species of bee introduced in the United States, and there are many, many species of native wild bees, and we really need to start paying a lot more attention to them, and I'll show you why. I'm going to talk a little bit about bee nutrition in general. So you all know this, that pollen provides the protein and lipid source for bees. And I'm going to show you these photos. This woman, Heather Holm, is a landscape designer in Minnesota, in the Twin Cities area. And she just loves native bees, and she's an amazing photographer. So the photo on the left, this flower, Dahlia purpurea, is purple prairie clover. It's a native flower. And it's nice to see a honeybee on a native flower like you see on the left. And look closely because there's a little petal on her pollen load from the flower. Can you see that? It's a cool, adorning her food, right? And on the right is a bumblebee on the same kind of flower. So you can see their pollen loads on their back legs. And then a couple photos of a honeybee with her proboscis, her tongue inside of a flower. I don't know left or right, so that side. And a little Colonies, a little native bee, wrapped around, hugging an onion flower with her little tongue in there, sucking the nectar out of that flower. So nectar, of course, provides all the carbohydrates and amino acids and vitamins and minerals, and I'll talk a little bit more about that. Just a few more photos, because I want everybody to pay attention to our wild bees more. So look at this little serotina on that side. Thank you. Oh, pointer. With her proboscis, right, gathering nectar, and the melisodes with her big loads of pollen on her back legs. And agaposmin, beautiful emerald green bees collecting pollen and probably nectar from the aster and anthophora. And I just love this photo. Look at the tongue, the proboscis on this bee. And then look at her legs. This is a campanula bellflower. It's like hanging on, getting the nectar. So when you start looking at these bees up close, it's a little bit like what Bill Cunningham does in the streets of New York City. You guys follow this? If you don't follow Bill Cunningham, go on the New York Times and look at he sits out in New York City and takes photographs of people walking by in all of their fashion, whatever they have on that day, and everything's marvelous and everything's beautiful. And when you start looking at native bees, on all the bees on the flowers, it's a little bit of the same thing. So honeybees, of course, collect a lot of nectar and pollen. They convert the nectar to honey, which we know about. And honey, this is research that was done by Dave Birnbaum's group at the University of Illinois a couple of years ago. And the title of the article is talking about how when there's some constituents, some components in the honey that upregulate genes that can help bees detoxify pesticides and help their immune system. And they identified these 3 compounds, which you don't need to remember, but actually those compounds originate in pollen and propolis, the resins that bees collect, and they're getting into the nectar. And so this one, P-coumaric acid, this first one, this is one they really looked at very, very closely. So that it comes from the bees' food, their pollen and their nectar, that gets into the honey. It actually turns on, this is the genome, right, the DNA, and genes make proteins, and some of those proteins are enzymes. Some of the enzymes they make are these things called P450s, which are basically, as Reed Johnson would call them, little Pac-Men. They are able to metabolize and break down some of the pesticides that bees collect. So think that through. So there's components in the bee's natural diet that helps turn on genes to make these enzymes to metabolize some of the toxic compounds that the bees run into. It's pretty cool. All right. This is Andrena, and in Minnesota right now, Andrena is just like erupting from the soil in many, many places. So they hibernate all winter long in underground all by themselves, and they come out from these little holes, and they're gregarious. So there's places where they all emerge basically at the same time, and it's happening right now, or just about last week. It's very cool to see Andrenas and other bees too. And they go to flowers. Right now our fruit trees are in bloom, and they go to those flowers to collect a little bit of nectar and some pollen, and they dig a new little nest hole, and they put their ball of pollen on there and lay an egg, seal the cell. So when that egg hatches, this is the little larva that's going to eat that pollen directly. And I'm bringing this up for a reason. And then that larva then will pupate and as an adult spend the whole winter in Minnesota underground. So most of their lifespan is underground. And we're trying to help people understand don't mulch the heck out of the top of the ground, because the these bees, it's not good for them. We need to have bare exposed nesting ground for these kinds of bees. They're really sweet little bees. Okay. But honeybees, when they collect pollen and they store it in the cells like this, the adult bees eat the pollen, the young adult bees. And then the young adult bees, when they eat that pollen, it stimulates the production of a lot of a storage protein and a yolk precursor called bilirubin. And in reproductive insects, this protein would go to help make eggs. But in worker honeybees, what it does is get shunted to the head to have their brood food glands develop. And so then their brood food glands are what are producing the food for the larvae, like this bee is feeding the larvae. And so the difference between honeybees and many of our solitary native bees is this larva is eating the pollen directly. And in honeybees, it's shunted through their bodies where if there is a contaminant, it could be detoxified first before they're feeding the rich brood food to the larvae. So when you see articles like this that came out a couple of weeks ago in Nature from Sweden about the seed coating around the neonicotinoid insecticides negatively affecting wild bees, and if you read through it very carefully, you read the effects on honeybees were not as noticeable, or if at all. That's why. This is why. Because our native bees are eating the stuff directly. They're eating the contaminants right in the pollen. And in honeybees, it's being shunted through the adult bee body, through glands, potential for detoxification through the adult bee, not necessarily through the larvae of the native bee. This gives us really good reason to think about all of our bees, not just our honeybees. And we need foraging areas for our bees to detox. They need good natural food to stimulate those detoxification enzymes. So there's a lot of movement to ban particular insecticides, the neonics, for example, and maybe that's one approach. But another approach that we should be doing at the same time is if we can provide all of our bees enough natural food, enough good, clean food, they can help detoxify some of those low-level compounds, low-level toxic compounds that are in some of the flowers. So one project that we're doing in Minnesota, in addition to trying to improve the landscape, we have a lot of lawn in Minnesota. You guys don't have that much lawn here, but lawns in Minnesota are big. They've always been big. And you're a good person and a good neighbor if you have a nice lawn, a nice tiny lawn. Well, I burned mine down 3 years ago. I burned it off completely and put in a prairie in my front yard. I have had some citations from the city of St. Paul. But I put up a sign from Xerxes that says pollinator habitat, and now it's a project and people are leaving me alone. This is one of my students, a master's student. The state of Minnesota, our lottery money, some of our lottery money goes into this trust fund. It's the Minnesota Environment and Natural Resources Trust Fund. And you can apply for grants from this. And I did one day after I got really sick and tired of looking at lawns around the Twin Cities. And I put in a proposal that we need flowering lawns. And this particular trust fund only funds things if you're promoting native species. So I said, all right, we'll look for some native flowering plants that you could seed into turf and that would continue to bloom even after mowing. It's a difficult project because a lot of our native flowering species need to get higher than 3 inches of lawnmower height to continue to bloom. But we're finding some potential species here that will continue to bloom. We were also looking at some non-native species on the side. Don't tell them. And we're going to be comparing bee visitation rates relative to regular Dutch clover that is already in some of the lawns. So we're just trying to enhance some of these areas that are dominated by a single species in our urban landscapes as well as our agricultural landscapes. And I want to talk just very briefly about Matthew Smart. He just defended his PhD, so he's Dr. Smart now. His thesis was this. He asked this question: how does agricultural land use affect the survival of honeybee colonies? And he did this in collaboration with the beekeepers at Browning in North Dakota and the Prairie Potwell region, big beekeeping area where a lot of beekeepers take their bees for the summer months. And so using Zach Browning's colonies and working with the USGS out of Jamestown, Minnesota, or North Dakota, they helped characterize the landscape in a 2-mile radius around where the apiaries were. So there were 6 different apiaries, and this one I just circled because site 6, one of the apiaries, and these apiaries had, 36 colonies in them, I think. It had, all the colors show that it had more potential flowering plants around in the 2-mile radius for the honeybees. In contrast, site 3, which was dominated by corn and soybeans and some wheat and other crops and other that are not flowering plants for bees. And then the others were intermediate in the availability of flowers around them. And over 3 years, as the colonies were in North Dakota in the summer, and then Matt followed into almonds and then back, and he followed individual colonies over time. Although the colonies were requeened every year, we just followed the colonies for 3 years. And he took all these measurements of the colonies and also took individual bee measures, including that bilirubin protein. And just to cut to the chase, adding up the survivorship of those colonies over the 3 years, this is actually how many were lost. And on average, over 45% of the colonies were lost at that site 3 over the 3 years. In contrast, it was about 12% on average at the site with more flowers. And I want to say that the beekeeper was very good at controlling his light levels, nosema levels, and there were no differences in any of the pathogen levels between the apiaries. And we also, before I go into this list over here, I want to say that we collected pollen from the colonies over a 3-year period. We characterized the pollen down to taxonomic level as best as we could, and we sent all of the pollen in for pesticide residue analysis. And over the 3 years, there was not any trace of any neonicotinoid in any of the pollen over 3 years. We found lots of organophosphates, we found lots of pyrethroids, a huge amount of fungicides, but we didn't find any neonics. Pesticides, when he ran a big model to see what factors most affected the bees' survival over these 3 years, pesticides were not in the picture in this particular study. But how much brood they had going into our fall in Minnesota or North Dakota, how much pollen they brought in over the course of the summer, their mite loads at the end of September, and then these, which I'm going to go into in the next slide, were most important in predicting the health and survivorship of the colonies. And those individual bee measures were really interesting because those are like a blood test in some way, analogous to a blood test that you go into the doctor and have your blood. They look at cholesterol or blood sugars in your blood. So if we take samples of young bees' blood, if in September their levels of bilirubin were really high, that was really the best predictor of how long those colonies would live. And it also was an indicator of the quality of the nutritional landscape around the bees. So it's amazing that we could get so much information on the one compound. And also their lipid levels were very good indicators. If they had higher lipids, they would survive longer. And if their immune system was not as activated in September, that was better for them. So if the immune system's activated, then it's harder on the bees. It takes a toll on physiologically on the bees. There's a great thesis. He's going to be replicating this now with USGS as a postdoc on a much larger scale across many other states to see how robust these measures are. All right, so I want to move on a little bit to other studies that we're doing in my lab, and they're all on how colonies defend themselves naturally. This is my my favorite area of study is looking at how bees can help themselves. And so when you start looking at a colony, there's an individual bee, of course, and they have their immune system. And then when they start behaving among individuals, their behaviors can be analogous to cells in the immune system. So this is a photograph of hygienic behavior where bees have detected a pupa, probably infested with a varroa mite, and they're removing the pupa from the cell. This is a picture of bees grooming each other. Those are some of the self-defenses that bees have. And as a colony, when they start doing these all together, it becomes really remarkable how they can help keep themselves healthy. And then there's one more layer. This is like layers of an onion. This layer, this is the propolis layer. And if you go into a natural tree cavity and you look inside the tree where bees are living, you'll see all around the nest cavity is this layer of propolis that they deposit naturally. And they get this propolis, it's just resin from some trees. And I'll talk about which trees, at least in our area, coming up. So this is a cottonwood leaf bud, and it secretes resin. It protects the leaf from infection. These resins are highly, highly antimicrobial, meaning they have antibacterial, antifungal, and antiviral properties. And the plants secrete them as secondary plant compounds. And bees go and they scrape these sticky resins off the leaf bud, and they pack them on their hind legs, very shiny. If you see them coming into your colony, it's resin, not pollen. And then they take them back to the nest. And this is a cutaway view of inside of a tree cavity, and that dark stained area is like varnish where they've deposited all those resins. Once it's in the colony, we call it propolis, or propolis. Tomato, I don't know. I'll call it propolis, and it's basically just the same resins. Sometimes they add wax to it. But it's, as far as we know, they're not modifying the chemistry of the resins as they come from the tree. So in our commercial boxes, of course, they put the propolis exactly where you don't like it, where you don't want it. It sticks everything together, and most beekeepers don't like it. You have to get it on your clothes. It never comes out. I want you to start liking this stuff. Because it is so good for the bees, but we're finding out. This is looking, if you're a bee in your house, in your tree cavity, and you're looking out your front door, this is how much propolis is deposited right around the entrance. It's very thick and important to them. So 3 different students have been asking these questions. How does the envelope, we'll call it that varnish or that envelope within the nest, how does it help the bee's immune system? Does it help them fight off disease? Where are these resins coming from? Are there some that are better medicines, if you will, than others? And do bees, if they're sick, do they collect more? I'm going to go through this very, very briefly. This is Renata Borba, who's finishing her PhD this year. She took off from where Mike Simone Finstrom left off. I'll just explain what she did. She went and cut up all our good propolis traps and stapled them inside the boxes to encourage bees to make a natural propolis envelope. We wanted to see how it affected their immune system over a long period of time, over a full beekeeping season. So she started bees up in April, ran them for a year. A new set of bees and new equipment, ran them for a year. And if you remove these propolis traps, this is what the envelope looked like inside. Okay? So this was our quote, natural propolis envelope. And she took lots of assessments of the colonies over time, the colony strength and levels of diseases and viruses and immune system activation. And the propolis envelope really had no, except for this is the first year measurement of colony strength, and by May of the following year, there was a significant difference. Colonies in the propolis envelope were stronger, but it wasn't true the next year. They weren't worse, which is good, but it didn't seem to really affect, at least over 2 years, over both years, the colony strength. There were no differences in levels of varroa, levels of nosema, or even in level of viruses. I thought they might affect the level of viruses, but no, they were all the same across those 2 groups. But if you start looking at their immune system, it was highly significant. So by the first fall, from the first group of colonies, their immune systems of the colonies with the Propolis trap was always significantly lower. And across many, all of these strange words are some of the proteins, the antimicrobial peptides that bees produce as part of their immune system. And most of these are cellular defenses. We have some humoral defenses and some transcript pathways. I mean, it was really, really robust. And this was the second year. Again, at least these two genes, the rest of them were not as significant as the previous year. And having a lower, or I'll call it a quieter immune system is a good thing. Okay, you don't want your immune system activated. So you want these bars lower than these from colonies without the Propolis envelope. You want to see the levels of these immune gene transcripts lower. But by the next year, and I'm just showing you May 24, it was the same for May 23. By the next spring, so now you have to remember, we're in Minnesota, and the bees winter there, starting in about November. We get a lot of snow, a lot of cold weather, and then they're not foraging, they're not bringing in any food, they're not bringing any resin in until about end of May, early June. So she looked at the colonies in May, and now no differences in immune gene transcripts between the two treatment groups. And so she collected some of the Propolis and did some analysis to see how active it was. And by the following spring, before the growing season, the Propolis inside the nest had lost a lot of its activity. Until the trees start growing again and the bees start collecting Propolis, they'll bring in fresh Propolis, and then the activity will start up again. So that was really interesting to see that over time it does lose activity, and you need to let the bees continually bring in more. Another part of her research looked at whether the Propolis was actually active against pathogens. So this is Mike Simone's part of his work, he challenged colonies. He made them sick with chalkroot, a fungal disease, and found that colonies with the Propolis envelope had a lot fewer chalkroot mummies compared to colonies without an envelope. And Renata continued with that, and she looked to see if she challenged colonies with American foul brood, a bacterial pathogen, what would happen with colonies in a Propolis envelope or no Propolis envelope. And this data shows that colonies without a Propolis envelope, this is opening up every cell at the end of the season to see how much American foul brood was in cells that were opened up already and cells that were still capped over. She opened them all up and found very highly significant lower levels of American foul brood in colonies with a Propolis envelope. So Mike Simone asked, do bees, if you make them sick with chalkroot, if you challenge them with the chalkroot pathogen, do they increase their resin foraging? Are there more bees that collect resin? And so what you do for this, and what we've done for this experiment, is sit in front of colonies and count the number of resin foragers coming back. You close up, these are little tiny 5-frame nukes. We cork the entrance, and we sit there and count how many resin foragers come back within 15 minutes. And you do this enough. You have to do it a lot. Because resin foragers are not very common. So in order to get your sample size high enough, you spend a lot of time sitting there. It's pretty relaxing. So here's a bee with resin, and here's a bee with pollen, and your eyes have to be good enough to differentiate them. And in fact, Mike found that this is, well, I won't go into this axis, but he found that you look at the resin foragers before the challenge, you make them sick, and then you look at the resin foragers after challenge. And so this is the difference, and you can see after challenge, there are more foragers that are collecting resin. Renata repeated this. She also challenged them with American foul brood, and again, graph shown slightly differently, but again, very highly repeatable. When you challenge a colony with chalk brood, after they are sick, they bring in more resin. There are more resin foragers. But that is not the case with American foul brood. It's fascinating to figure out, okay, now we have to look at the mode of transmission between a fungal pathogen and a bacterial pathogen. They're very different. One is on the body. This one goes inside the body. And so how come they may not notice, or what's the difference? We don't know. That's where we're at with that study. So they do collect more resin if they're challenged or if they're sick with a fungal pathogen. A different student, Mike Wilson, started looking at where are the sources of resin. So he got his PhD in plant biology and horticulture, and he was working with some amazing plant chemists in that department, and he did metabolic fingerprinting. So he would take samples of resin directly from the bees' legs and from trees and run them through an LCMS, liquid chromatography mass spectrometry, and he could get this fingerprint of resins. It gives you all the compounds. And then you match them up from the tree with what's on the bee's leg, and you match the fingerprints, and you can tell what the bees are collecting. So he climbed all these trees around campus, and that's what he got to do for a PhD, and collected all these resins from all these trees. And what he found is the bees are mostly collecting from our cottonwood trees. They're not the most prevalent, but they're the ones that they're choosing to collect propolis from cottonwood in our area, and some from balsam poplar. And then he takes those into the laboratory and does some tests to see how antimicrobial they are. And so in this case, we have culture medium that's inoculated with American foul brood bacterium, so Pennicillus larva, and then you put propolis into the culture, and you can see how quickly the propolis acts as a quote antibiotic to clear the bacteria from the culture. And you can measure this optically with the spectrophotometer. And in fact, Propolis deltoides, this is cottonwood, this is the trees that he collected on campus, and the ones at the top are the most antimicrobial, the most antibacterial activity against American foul brood, and it turns out it's not cottonwood. The cottonwood is kind of at the end of the list. They were all really had good properties, but white spruce was at the very top, and yet these were not collecting from white spruce. It's very curious. So there's lots of information in this, and lots we don't know. It's just the tip of the iceberg. It always happens when you ask bees a question, they give you an answer. It's like, oh God, why can't you just say this one was the most active? It used to be black and white, but it's kind of where we're at with these studies. Okay, I'm going to wrap it up, and yes, Dr. Perella, Mike Perella was right. We are building a new bee and pollinator research lab. We're going to be breaking ground this August. This has been a long battle for me because unlike the Davis facility, I've been working out of a garage since forever, since 92. We have a lab in the entomology department, but it's been very, very difficult. So our state, the state of Minnesota, finally, the university put forward a capital request to build a new facility, and the state put in the 2/3 funding in their bonding bill to have it built. And so it's going to have offices. This is not the greatest. You don't get the full impression from this particular design. We're just at the end of the design phase. But this half of the building behind there is the labs, 3 labs, a technical lab and 2 practical labs and office buildings. And this side, coming in here, is garage, extracting room, hot room, shed. I even convinced him to give us a shower. I won't tell you the argument I went through. This is super exciting, and actually Eric Lesson sent me the blueprint, the floor plan from the Davis lab, and we use that a lot in the planning, so thank you very much. Really appreciate it. And so in this building, I'm also really happy to announce we were just able to hire a new faculty person, Dan Carroll, who's doing a postdoc currently at Rutgers with Rachel Winfrey, and he'll be coming in August, at the end of August, and he will be our native bee person. So we're trying to compete with Davis. We'll never catch up, but we're trying. And so really super, I am so relieved. You can't imagine how relieved I am to have another colleague to help study the native bees. I'd like to get back into some honeybee breeding, and this will allow me to do that, dance or Another thing that happened in the last couple of years is some people in town in the Twin Cities donated a lot of money, $2 million, to build a public facility to showcase bees and pollinators. And this will be at the university's Landscape Arboretum, which is about 30 miles west of the Twin Cities. It's in this huge, beautiful, beautiful area. And in one of the areas, there's an old red barn. And they're going to build this public facility. This will be for school kids and for anybody that goes to the Arboretum. And it will have lots of exhibits about honeybees and native bees and a lot of landscaping, which is not totally shown here yet in this schematic. They're just also finishing their schematic design. And once they have this building, they're hoping to build all the way around it food, like an urban agricultural center, so that you can learn about how to grow food, how to process food, how to cook the food, all in this area. And the bee center, the bee discovery center, will be the center of the first building that's going to be built there. So we have a lot going on now. It's super exciting, and I have a lot of meetings with architects, which All right, so I want to end now. I'm just going to say thanks to, I have a lot of graduate students, a lot are in the middle of, have graduated or are graduating this year. A great group of students. And I want to thank all of you and funding sources. And thanks for having me.
Right, we have about 10 minutes for questions. We have a roving mic, so please raise your hand. We'll see you get a mic so everybody can hear the question.
Hello, thank you. So does the propolis level indicate in the hive for practical beekeeping that there's more disease present?
Good question. So more propolis, does that mean more disease? No. This means it's good. The bees it's a genetic trait, right? So some bees will collect more resin than others, and you can select for more resin collection in colonies. It's heritable. So having a lot of propolis in the nest is a good thing, and you can look inside and see if there's more disease. Probably not. And it would only be for chocolate as far as we know anyway.
I don't understand if pollen can help detoxify pesticides. I thought I read where the neonics are expressed in the pollen, and that's what is a problem for the bees. So how does that equation work out?
Right. So that's why they need good clean flowers, those that are not contaminated with insecticide, so that when they do eat flowers that are contaminated, they're able to detoxify. So if they don't have the good flowers without the contamination, they need the good flowers so that they can get P-coumaric acid and other compounds in there to activate their detoxification systems so that they're able to detoxify some of those insecticides. Now, I don't know exactly if the P450s detoxify the neonics, but it's just an example of why we need good, clean bee food. Does that make sense? I didn't say they weren't a problem. I was just saying they're not always the problem.
So you said that the propolis becomes less and less active over time. So as beekeepers, do you think it's better to scrape that propolis off so that they recollect, or do they just build it on top of the propolis that's already there?
Okay, so it depends where you are. If you're in California, the bees are probably going to collect resins most of the year, so it will continue to have activity if there's plants around that are blooming or growing, let's say, okay? So our Minnesota winter, right, they're not collecting any resin from October through almost June. So over that period of time and through our severe winter, the propolis inside the box is seeming to lose activity. But if your growing season is extended in most areas of the world, and it is longer than that, then I don't think you need to worry about it. You can scrape it off and let it collect more.
But they'll layer it.
They will layer it, yeah. Oh, yeah.
How much of a problem are neonics for honeybees? And in what way?
On a scale of 1 to 10.
And in
I don't
a
need much short. This is such a complicated issue. Neonics are one kind of insecticide, and you know how many kind of insecticides are out there. When you look at what's in the pollen of bees and you go, oh my God. And it's not just insecticides, it's herbicides and fungicides. So how much of a problem are the neonics? If they run into a high enough dose, yeah, they're a problem. But if they run into a high enough dose of organophosphate or a pyrethroid that are sprayed on soybean aphids all the time, that's a problem. So overall, how big of a problem are the neonics? It's hard to say. If they get into a high dose, yeah, it can be a problem. If they get into chronic low doses, we're starting to understand how big of a problem it is. For native bees, probably big problem, if that's their source of nectar and protein, the pollen. Okay? What the neonics are for me is an entryway for people to start getting engaged in what's going on with pesticide use in this country. So if that's your starting point and you want to grab onto a neonic issue, go for it. Push it as far as you can because it opens the conversation into what we need to, the big correction that needs to happen in how we're using pesticides and how much and where. So we really need pesticides in some cases and our pollinators, and we need to figure out how to use these pesticides more judiciously. So the neonics are a problem because of their persistence. That's their problem. And their complete disregard for any integrated pest management of treating when it's a problem because they're always there. Okay, but are they the biggest problem for bees? Not necessarily. Okay, that was a soapbox.
I was wondering, should we be planting trees that do the propolis?
Yeah. Actually, we found, I forgot to mention, we collected propolis samples from other parts of the United States. And one of them that we got from Propolis fremontii, which is a cottonwood out of Nevada, which you may have here. The antimicrobial properties in that was much higher than anything we had in Minnesota. And in fact, the chemists went in and looked at compounds. They found individual compounds that are extremely active, and actually they're new antibiotics. And they're working with the patent office in the university to patent these compounds as antibiotics for bees. We haven't tested any human stuff. But the potential is amazing, and so yes, plant trees.
Could we get a list of which are the highest?
Well, we don't know for California, right? We know on campus. Propolis fremontii, if you can get that here, is a good choice.
Could you repeat the name of that?
Does anybody know the common name? The plant cottonwoods, native cottonwoods. Yes. Yes.
Are there any studies being done that you're aware of? Are there any studies being done that you're aware of the how bees handle the resins in the hive to, create the propolis and apply it?
Yeah, it's fascinating. Tom Seeley and a postdoc from Japan did this really nice study, and there are descriptions, and you can see them. And so when the bee has the resin on her back leg, often she can't even get it off of her leg herself. Another bee has to come pull it off. You ever seen that? It's kind of amazing. And so then the bee takes this sticky resin, and then they use their mouth parts to stick it in parts of the nest. Tom Seeley and this postdoc, Nakamura, found that, in fact, the bees were doing the dance language sometimes to recruit other bees to the propolis sources. That's an area that needs a lot more study. It'd be really fascinating. Please tell me when to stop you.
Would there be merit to rough the inside surface of a hive body so that it would be coated with propolis? And also, would there be merit in removing the propolis from the frame rest and then dissolve it and reapply it manually?
Yeah, so there is merit. You can rough up your hive boxes, but it would just be better if our beekeeping manufacturers would just sell unfinished lumber inside the boxes. I believe John Miller, wherever he went, made some of these or had somebody made, and I don't know how those are doing for him. We can ask him when he comes up. But yes, it would benefit. I think it would be a good idea. Manually roughing it up is hard, and it doesn't get him as rough as really unfinished lumber. And should you scrape it out of where the frame rests are, yeah, you can. But it's, collecting the propolis, dissolving it in ethanol, getting the right grade of ethanol, the right percentage, and then applying it back to the colony. I'd just rather let the bees do all that stuff. Better at it.
So in this, the idea of the good clean food for the bees, the local jurisdictions could have a role to play, and I think some are becoming bee-friendly cities or so on. What is your experience with local jurisdictions trying to impose regulations or encourage decreased use of pesticides and the increased planting of the safe clean food for bees?
Yeah, so Minnesota has 2 cities now, Stillwater and I think it's Edina, that are considered bee-friendly, where they were very successful with their city council in eliminating pesticide use in those particular areas and planting more bee flowers. So at least where I'm living and where I stand, there's a movement in that direction and it's been successful. So Mike's giving me a gap. He says, one more question, and there was a guy in the way back. Oh, sorry. Okay.
Can you explain why a strong biological response is not good for the bees?
Okay, think about it yourself. If you've got an allergy to something, your immune system's going to be chronically cramped. And that's not good for you. What you really want is to get rid of the allergy, right? So your immune system is not cramped. When your immune system is activated, it's at the expense of something else. And studies in insects shows that it's at the expense of survivorship. They don't live as long. Sometimes at the expense of honey production for a honeybee. So that's at a cost. There's going to be a trade-off. And I'm going to take one more because he was really nice.
Marla. Collaboration potential for European countries because you have the much larger climate range and a shorter distance. And I don't know how the migratory practices in Europe compared to here, but then you have a better chance of comparing to the northern climate versus the southern desert climate in Europe. It's like we're all desert here. This is almost all desert, but then the bees come in, the bees leave. How does that compare to the propolis collection?
So the propolis in northern Europe is from poplar trees, and they've known that. People in Bulgaria are doing a lot of research on propolis, always have. And so, yes, it's very comparable to what they're doing in Europe. Does that answer your question? Oh, I don't know that there have been that many studies in southern Europe. Okay, thank you very much.